afternoon, everyone. My name is Mike Nielsen, and I work with Nelson Laboratories. We're a contract testing, microbiology testing laboratory, and I'm going to be talking today about reusable medical device validations, reprocessing validations. This is a hot topic right now with the FDA and the reusable medical device industry. So I'm going to be discussing some of the trends that we are seeing right now, and hopefully answer some questions that you may have regarding some of these validations and what we're seeing right now. So obviously, as technology continues to advance, we are seeing an increase in the complexity of reusable medical devices. Along with that complexity in devices, we are unfortunately also seeing increased difficulty in the ability to reprocess, clean, disinfect, sterilize these devices in a hospital setting. Because of this, there's a lot of scrutiny right now in the industry. The FDA, as most of you are probably well aware, recently came out with a draft guidance document for reprocessing reusable medical devices. This was released for comment in May this year. And this document dealt a lot with these validations that I'm going to be discussing today, as well as gives some advice and direction to manufacturers for what they need to include in their instructions for use for the end user. As well, we see that AMI is doing what they can to try and keep up with the industry expectations. 2010 was the most recent revision of TIR 12 that has come out, as well as TIR 30, which was this year that the 2011 revision was released. So my presentation today, I'm going to be discussing some of the new FDA guidance and trends in reprocessing validations. I'm going to be talking about manufacturers' responsibilities along with these trends. I'm going to talk a little bit about how this all relates to instructions for use, and then I want to discuss some of the areas where there is still need for continued focus and unfortunately some holes in how we perform these validations. So first of all, I'm going to focus on cleaning validations. This is the first step of the decontamination process. Definitions out of TIR 12 are to render reusable medical devices safe for handling by healthcare personnel and to prepare reusable medical devices for additional reprocessing. Obviously, if you have residual contaminants on your device, this may interfere with the efficacy of further processing if you're going to be doing disinfection or sterilization on these devices prior to the end use. So device condition is a topic that is causing a lot of problems. Historically, device validations have been performed on brand new devices. This is a first step prior to receiving FDA approvals and being able to market your device. So typically, used condition devices are not available from the manufacturer when these validations are being performed. However, used condition devices are critical to understanding how effective the cleaning, disinfection, sterilization can be performed on a device that has been in the industry, that has been reprocessed several times. So how do we get used condition devices in order to do these reprocessing validations? Well, we simulate use. We take the devices through a series of decontamination cycles through repeat processing. So this typically may look like this, with manual cleaning being a component. Maybe it would go through the automated washer followed by sterilization. So a series of cycles would be performed in order to simulate a used condition prior to the start of the validation testing. The next piece to the puzzle is understanding how many device replicates as well as process repetitions we need to take these devices through in order to be considered a full validation. The FDA has been very vocal that a minimum of 3 test devices should be used. This has been industry standard for a long time. However, the number of process repetitions that we take these 3 test devices through has recently been catching manufacturers off guard, by surprise. Typically, one process rep of 3 devices constituted a validation. Now manufacturers in their submission process are being asked at times to provide more process repetitions than the single rep. What number is being asked for? Well, the FDA was not giving specific numbers. They are saying that the manufacturer needs to justify the number of process repetitions that are adequate to demonstrate reproducibility. So I put this slide in here just to take you through the cleaning validation process a little bit, familiarize yourself with the process, and then I'm going to take you through different steps of this process and what the trends are with the FDA currently. So typically the devices would get contaminated, then cleaned, then after they have been cleaned, the devices are extracted in an extract fluid, and it's the extraction fluid that then gets tested for residuals that may be present still on the device. So where can we submit worst case testing into this process? Well, the first step is by choosing a clinically relevant soil. So when we're looking at test soils, the FDA wants you to look at the end use of the device. What is the device going to be in contact with? Is it going to be in contact with circulating blood? Is it going to be in contact with tissue, bone, etc.? And then you need to choose a soil that represents those conditions that the device will be seeing in the clinical setting. These are some of the standard test soils that we use in our laboratory. We have blood-based soils. Defibrinated blood soils typically are our go-to blood-based soil for any devices that will be in contact with blood. Hucker's test soil is often used to simulate feces contamination. Miles test soil is a test soil that's specifically used for any devices that are going to need LAL testing, endotoxin testing. We've noticed that our defibrinated blood and other blood-based soils have interference with the endotoxin screening assays. So we have this specially designed soil specifically for that purpose. And then there's British soil, which is typically used when the device will be in contact with any mucous membranes. Unfortunately, these soils, they do have components that are clinically relevant. However, oftentimes these are missing key components. If a device is going to be in contact with tissue, with bone, how do we add those constituents to our test soils? The FDA is asking for this. However, we've noticed when we have tried to add some of these constituents, we are seeing inhibition with the assays that we are testing at the end of the process for residuals. So there is some problems that we need to still work through. Also, we have commonly been able to integrate some of these soils, a combination of soils, for this testing in order to achieve clinical relevancy. But the FDA wants to see clinical relevancy as it applies to constituents, amount of constituents, tenacity, and also viscosity of the test soil. So the next step where worst case testing should be applied is in the contamination of the test devices. So we need to choose methods that simulate worst case contamination. We need to contaminate lumens, hard to clean areas, cracks, crevices of these devices. We need to simulate use. If the device will be used in a surgery and be submerged for an hour or two, then we need to simulate that in our validation methods, actuating the device while it's submerged. The next piece to this puzzle is the dwell time. The hospitals have been given best practice advice to begin the decontamination process as soon as the device has been used. However, this is very rarely the case that this occurs. We have seen hospitals tell us that devices have sat contaminated for up to 24 hours even at times before they begin the processing. So the FDA is asking for worst case dwell times to be used as well. They want to see the soil dried onto the device before we go into the cleaning process. So here are some pictures just to demonstrate some of the contamination procedures that we do in our laboratory. Here's one of our analysts contaminating just some simple scissors, actuating these scissors so that the test soil gets into the cracks and crevices. And then here's some devices just sitting on our stainless steel counter to simulate the dwell time necessary before we start the cleaning process. So if that wasn't enough, we now introduce worst case testing into the cleaning steps as well. We want to use minimum times, temperatures, procedures to demonstrate effectiveness. We need to look at our choice of detergents. What materials is the device made of? We need to make sure that the detergent is compatible, that it's compatible with the constituents, the contamination that will be on the devices. We may question using an enzymatic detergent. pH range is certainly an attribute that we need to look at. But one thing that the FDA is pushing as far as the choice of detergent is they would like medical device manufacturers to use the minimum labeled soak time of the detergent in your validations, if possible. Why? Because of human factors considerations. In the hospital, if they don't have an IFU readily available, they're going to go off of the detergent labeled soak time. So the FDA would like to see this prior to any extended testing that may need to occur for more complex devices. So here's just some considerations to look at. I've already discussed the soaking, but also brushing, flushing. Want to make sure that we are appropriately getting this detergent into the cracks and crevices and hard to clean areas of the medical device. There may be mechanical methods using ultrasonic cleaners, automated methods using washer disinfector. And I've got a picture here of one of the automated washer disinfectors that we have in our laboratory that simulates what you would find in a hospital setting. Here are some standard parameters that are default parameters for many of the hospital settings. We want to make sure when we're validating these settings that we are using minimum circulation times, rinse times, and the low end of the water temperatures. Once the devices have been cleaned, they go through a visual inspection to make sure that any visual contaminants have been removed. Most of the time this is done with the naked eye. Some microscopic methods can be used. We are leaning more towards using scopes now for this process, especially with these lumens and hard to reach areas. And then the device gets extracted. There are different extraction methods. The most common is a manual shaking method where the device is submerged in extraction fluid and shaken over a manual path, 12-inch path 100 times. And it's a very good extraction method. However, there are devices that are not compatible with that method that can't be submerged, and we have to come up with alternate methods. So we do have swabbing options. We do incorporate sonication where necessary. But one thing that the FDA, one trend that we are seeing is they are asking for those extraction methods to be validated concurrent with the test using extraction efficiency. So how this happens is we take our positive control device, it undergoes the normal single extraction, but then we follow up by performing multiple subsequent extractions of that same device. And the efficiency is calculated by taking the initial recovery from the first extraction divided by the total recovery of all extractions. This efficiency then can be applied to our residual levels of our clean devices in order to give corrected counts. Here's an example of some of the cleaning markers that are commonly tested. This is not an all-inclusive list, but these are some of the more typical common markers that we see at Nelson Laboratories. Bioburden has historically been the go-to marker in this industry. Then we have protein, hemoglobin, carbohydrates, total organic carbon, detergent residuals, as well as MEM, endotoxin, lipid markers. There are several ATP is one that we're starting to see more and more asked for in this industry. So there are a lot of markers to help determine if your device truly is clean. Now, Bioburden, as I said, this is historically the go-to marker. Acceptance criteria for this marker, the FDA has wanted to see a 3-log reduction using spore and vegetative gram-positive, gram-negative organisms. However, here's our disclaimer. The FDA no longer is accepting bioburden reduction, as a viable organism counts cannot be directly correlated to device cleanliness. However, we are still recommending bioburden be done with our cleaning validations because this is the only historical comparative marker in order to compare your cleaning process with devices that have already been released to the market. Also, Europe and Canada, we've been told if you're trying to market your devices there, they still want to see bioload reduction. So here's an example of our bioburden process. Here we have an analyst plating extraction fluid and then plating the filter onto nutrient agar, and it would grow up and we would see what our recovered counts truly are compared to a control that was not cleaned. I'm going to take you briefly through some of these other slides. Protein, we use at Nelson Labs the micro-BCA protein test, which is a spectrophotometric assay. Hemoglobin, in order to get the sensitivity necessary for this marker, we do use a semi-quantitative method. Now, the FDA is asking for quantitative methods to be used. However, we have not found a method that is sensitive enough using a UV spec. So we make a standard curve using the UV spec, and then we perform dilutions of from that curve from known concentrations. We apply our hemosticks to those concentrations, and then we're able to get a semi-quantitative amount. We know that based on the color change of the hemostic that it falls within a specific range of concentrations. Carbohydrate testing is performed at Nelson Laboratories. We do endotoxin testing as well as total organic carbon testing. And briefly, just to talk about those marker tests, more and more common we have seen protein being used as well as TOC and carbohydrates, but the FDA does want to see at least 2 markers selected. One of the issues with these cleaning validations is that there are no acceptance criteria established for every medical device type. AMETIR 30 does offer benchmark criteria from research that was performed on endoscopes. However, these criteria may not be appropriate for every device type that is out there. So the manufacturer needs to predetermine or pre-establish their acceptance criteria for their device as it relates to the end use of the device and what it's going to be in contact with. So another trend that we are seeing is destructive testing. This is where you would take the device through a series of cycles again where you contaminate and take it through the decontamination process, and then after several cycles of this decontamination process, you would then take your device apart, lumens, cracks, crevices, hard to reach areas, and see if there is visual soil accumulation that is occurring on these devices that may be of a concern if it were to be released and have patient contact. So some of the areas of greatest concern are outlined here: lumens, hinges, cracks, crevices, springs, braided cables. These are all very difficult to clean items. So then I'm going to discuss a little bit about sterilization. This, fortunately, is an area that has come a long way over the last 20 years. We have many processes available: steam, STERRAD, EO, dry heat. The new FDA guidance document, the draft guidance document, does go over these different processes and does recommend standardized procedures that you should go through. Here's a bank of our steam sterilizers at Nelson Laboratories that we commonly use for this validation testing. So with sterilization, we must be consistent with what is happening in the clinical setting. Um, Manufacturers need to validate per the ST79 parameters that are listed. Also, any chambers, accessories such as BIs, containers, trays, wraps should be FDA cleared for the intended use. And then one thing that is FDA has recently been looking at strongly is worst case load configurations for the chamber. Is it enough to just validate your tray full of devices in the autoclave chamber, or should you have a loaded configuration? Would that be more worst case? So I wanted to just take you quickly through the 7 criteria outlined in the new draft FDA guidance document for reprocessing instructions for use. So they say that the labeling should reflect the intended use of the device, that the instructions should teach the end user how to thoroughly clean the device. You need to incorporate a microbiocidal process, so sterilization or high-level disinfection. Reprocessing should be technically feasible. Use legally marketed detergents and or disinfectants. Instructions should be comprehensive, even including reuse life testing if applicable. So this is simulated use testing over time to determine if your device is still functional after so many cycles. And then instructions need to be understandable. What a lot of medical device manufacturers don't understand is that the hospital setting in these central reprocessing areas, often your education level is not high. They need to be able to read these instructions and understand how to carry out these instructions, perform these instructions. So what are the next steps? Where are the holes in this process that still need to be looked at? Standardized test soils is a big concern. We need to have standardized test soils for device families, as well as standardized repeatable reprocessing steps. The industry is asking for more standardization as far as these devices and instructions are concerned. Standardized clear instructions, and then finally, established acceptance criteria for these device types. How do we get there? Well, right now, nobody really wants to take ownership of these areas of concern, but Amy is working on some of these things as well as academia. So, and that's really all I have for you. I just want you to know that Nelson Laboratories, we really take pride and ownership in partnering with medical device manufacturers to come up with the most appropriate validation plans and to help you succeed, to be successful at getting devices out that are safe and effective. Thank you.